again. All right, so that's kind of the background on epigenetics and some interesting phenomena. So now let's talk about DNA methylation. What is it? It's actually super boring, kind of simple and boring, right? It's a methyl group, CH3, teeny tiny little group, being put on cytosine, on the C5 position on cytosine, right? So this is catalyzed by DNA methyltransferases. So this is a very simple, kind of boring chemical reaction. You know, it's very, very teeny tiny group put on the cytosine. In human or invertebrate in general, this methylation usually happens on the CG dinucleotide contact or CPG. So this is what we usually call CPG, right? So it's so simple. However, by having this methyl group over this C5 position in CPG, the biochemical property of this part of the DNA changes dramatically, right? So without the CPG, without the methyl group, right? So the DNA is like open, you know, happy and being expressed. But with the CPG, the DNA or the chromatin becomes like, you know, condensed, repressed, sad, and, you know, stop, you know, not expressing, right? So biochemically, this is actually putting on a very big difference on the chromatin, right? So that we actually sometimes call methylated cytosine the fifth base, so A, C, G, T, and methyl C, because methyl C has very different biochemical property than the unmethylated cytosine. Okay, so we know a lot about the methylation process. Is categorized by DNA methyltransferase, and then this is sort of a history of how people figured out DNA methylation. So I'm not going to go over this, but if you're interested, there are a lot of literatures to enjoy, right? So for maintaining DNA methylation, or for putting down the DNA methyl, for putting down the methyl group, we rely on two types of DNA methyltransferases. One is called de novo DNA methylation transferases. So we use there are two in human, 3A and 3B. They can directly methylate a piece of DNA, right? But we also have another one called DNA methyltransferase 1, which is a maintenance enzyme. So what this one does is during cell cycle, right? So when the replication happens, the newly synthesized strand is not methylated, right? So DNA methyltransferase 1 can actually recognize that and then come in and methylate the newly synthesized strand. So this is how DNA methylation pattern can be faithfully inherited across cell cycle, right? So this is the foundation for this epigenetic inheritance because you can maintain this methylation pattern. So this process has been worked out, you know, many years ago. The reverse reaction, the demethylation reaction, took the field about 20 years to figure out, right? So this is really just figured out over the last 10 years, right? Turns out there are two ways for us to remove methylation. One is passive, right? So what I mean is, you know, I just mentioned DNA replication, right? So during cell division, if you make your second strand, right, but somehow you don't maintain it, right? So you don't have methyl groups, you don't have the enzyme, right? So during the cell cycle, if you don't maintain methylation level will reduce by half, right? So if this happens around, you know, for several rounds, very quickly you can lose DNA methylation. So this is how we demethylate our genome through passive mechanism, right? So this is cell cycle associated. But we actually go around and then spend a lot of effort to actively remove DNA methylation. So this actually argues that, you know, this must be very, very important because we spend a lot of trouble. So we do this, so there isn't a demethylase, right? Turns out this reaction is very thermodynamically unfavorable, right? So we go through a series of oxidation reaction followed by DNA repair, okay? So it started with methylated cytosine, you use enzyme called TAD. So there's 3 TAD enzymes to oxidate this to hydroxymethylation, to further oxidate to formomethylation and carboxymethylation. And then the cell will recognize formomethylated cytosine and carboxymethylated cytosine and use base pair excision repair system to scoop out the methylated cytosine and then patch in back a unmethylated cytosine, right? So this is sort of the active process of DNA demethylation. So it's pretty involved, which argues, it's probably important, right? Okay, so we're gonna quickly go over the normal pattern and function of DNA methylation, right? Turns out CpG methylation is not evenly distributed in the genome, right? So in our genome, as an example, about 70% of CpGs are methylated, and they're mostly focused in or concentrated in the repetitive elements or transposable elements, right? So they're, in fact, their main purpose of DNA methylation is keep those regions repressed, right? So CpG density in the genome is also very, very uneven. So we overall CpG is very, very depleted, except in the CpG island. So these are CpG-rich regions that are usually having important regulatory roles. For example, 70% of gene promoters are actually in CpG island, right? So these regions are often kept unmethylated. Turns out, when we see these are CpG island, CpG rich, it's really, they really have the sort of the expected CpG density. So when when a CpG is methylated, it's actually much easier to mutate, right? So mutation rate on methylated CpG is much higher than unmethylated CpGs, right? So the reason that we see CpG island is because overall, the genome, the CpGs are methylated, so they are mutated away during the course of evolution, except for regions where they're kept unmethylated, right? So these are promoters, regulatory elements, they're kept unmethylated, therefore they don't have an elevated mutation rate, so they show up as CpG island. In fact, if you just look at the CpG distribution in our genome, that reflect methylation pattern of germ cells, right? Because in germ cells, the methylated CpGs start to mutate away, and that's what passed on to in generations, right? So by looking at the just a genome reference, you can actually estimate the methylation level of germ cells. Demethylation has a lot of very important functions, even though there's such a simple modification. The most important, in fact, some people argue that this is the only important function for demethylation is host defense, right? So they're used to silence parasitic sequences like transposable element, like the IAP element that we talked about at the very, very beginning, right? So some people argue that's the only function and everything else is kind of a derivative. But regardless, demethylation plays many, many important roles. For example, imprinting, right? Imprinting is pretty important, interesting, right? We inherit 2 alleles of everything from our parents, right? So that's kind of building redundancy, right? But sometimes, one of the alleles get methylated and does not express, right? So that's a very common phenomenon in placental animals, right? So there are a lot of interesting theories about why we have imprinting. The most dominant or my favorite theory is this is actually how your parents are still having a fight inside you, right? So it turns out, dads are usually very selfish, right? So they only care about themselves and passing on the genetic material. So what they want is big babies. They want their baby to be like really powerful and big, right? So they don't care if the baby takes too much resources from the mother. But moms, they want the babies to be healthy, but she also wants to reserve some resources for herself and the siblings, right? So what do? So mom figure out a way. So mom can methylate the pro-growth genes coming from dads, right? So it doesn't matter how much dads want the baby to grow. Mom said, I'm gonna shut down your pro-growth gene, and I don't care what kind of gene you bring in anymore, right? So that's actually a theory about how imprinting was evolved. So that main mechanism is demethylation. X chromosome inactivation, right? So we, in human, females have 2 X's, male has 1. Different animals have a different way to balance the expression from X chromosome. What we do, what human does is in females during development, right? So this is early stage of development. The X chromosome, the 2 X chromosomes, one of them get randomly chosen and get inactivated through demethylation and long non-coding RNA, right? So as a result, females are actually all mosaics in terms of X chromosome expression pattern, because this happens in early development. All the daughter cells from that point on will express the same X chromosome. So as a result, some, maybe some, over here it's the dad's X chromosome being expressed, but over here is mom's X chromosome being expressed, right? So demethylation is used to establish this X chromosome inactivation. Demethylation is also important for heterochromatin maintenance, chromosome stability, telomere length, and all of this. And also importantly, gene expression control, right? So we're going to go over some of these in more detail. But before we do that, I want to still describe one of the most fascinating process relating to DNA methylation, that is reprogramming, right? So DNA methylation pattern got reprogrammed twice in our lifetime, right? We kind of know the process pretty well, but we still have a ton that we don't know. We don't know how that's established. We don't know how the specificity is achieved. But let's just go over this interesting pattern. So starting from germ cell formation, right? The first thing the genome does, the cells, this germ cell does, is to remove demethylation. So everything gets wiped out. So this is probably how life starts with a clean plate, right? Rather than get everything inherited, we start with a clean plate. So now germ cells will develop, sperm and oocyte. So they establish their very cell type specific demethylation pattern. But when the germ cells, when sperm meets oocyte and form this zygote, right? So the first thing these cells do, right, is to remove demethylation again, right? So this is a second wave of reprogramming, and people believe this is actually how we reestablish this topipotency, right? So this epigenetic potential is reestablished by this reprogramming process. But interestingly, the paternal genome and the maternal genome undergo DNA demethylation in different ways, right? So the paternal genome actually goes through active demethylation. So this is where TEP enzyme is involved. You actively remove methyl groups. But maternal genome goes through this by passive demethylation, which is associated with cell cycle, right? But by the time of pre-implantation, right, the genomes of these cells are all demethylated, except for some imprinted control region. So this is where the previous generation may have some impact on you, right? So after implantation, development start, this demethylation pattern gets faithfully reestablished, right? And then the cell type specificity is often established during this process in development, right? So we know by and large, this is what happened globally, but we don't know the specific mechanisms behind each step and how the specificity is achieved. So there's a lot of interesting stuff that we still want to investigate, right? But in the context of what we want to understand of this normal pattern of demethylation is in relationship to gene expression and genome stability, right? So here, I use this to represent a euchromatic region, and over here is this paracentromeric region, right? So the little ticks here represent CPG site. If there is a lollipop on top of it, this means that CPG site is methylated. Over here, I'm drawing 2 genes with 2 exons here, 3 exons here. So typically in normal situation, you see higher CPG density in promoters, and these promoter CPGs are usually unmethylated, right? So this actually allows transcription to happen, right? It doesn't like guarantee transcription, but this is sort of allowing transcription to happen. So in intergenic region and in gene body, right? So these CPGs are usually methylated, and in intergenic region, you actually see reduced CPG density for reasons that we just talked about. In contrast, paracentromeric regions, these are full of like repeats and things like that, right? So these CPGs, these regions have low CPG density compared to CPG island, but the CPGs are all methylated, right? So this is actually really important because these regions have a lot of repetitive sequences, right? So these repetitive sequences are perfect subjects for non-allelic recombination, okay? However, DNA methylation repress that kind of recombination. You can imagine if you lose DNA methylation, these repetitive sequences start to recombine and your genome becomes fragile. This is actually how chromosomes get broken, right? So this DNA methylation is important for the stability, okay? All right, I think this is a good place to restart, to